Bismillahirrahmanirrahim and Assalamu alaikum. Well, my name is Iman Zahid from the Department BS Biotechnology, and the topic of my discussion is scanning electron microscope. First of all, microscope is the technical field of using microscopes to view objects that cannot be seen with the naked eye. There are three well-known branches of microscopy: optical, electron, and scanning probe microscope. Optical and electron microscopy involve the diffraction, reflection, or refraction of electromagnetic radiation or electron beams interacting with the specimen and the collection of the scattered radiation or another signal in order to create an image. Scanning probe microscopy involves the interaction of a scanning probe with the surface of the object of interest. Electron microscopy. An electron microscope is a microscope that uses a beam of accelerated electrons as a source of illumination. As the wavelength of an electron can be up to 100,000 times shorter than that of the visible light photons, electron microscopes have a higher resolving power than light microscopes and can reveal the structure of smaller objects. Electron microscopes are used to investigate the ultrastructure of a wide range of biological and inorganic specimens, including microorganisms, cells, large molecules, metals, and crystals. Industrially, electron microscopes are often used for quality control and failure analysis. Modern electron microscopes produce electron micrographs using specialized digital cameras and frame grabbers to capture the image. Types of electron microscopy. There are two types of electron microscopy: scanning electron microscopy, also known as SEM, and transmission electron microscopy, also called TEM. First of all, scanning electron microscopy. A scanning electron microscopy is a type of electron microscope that produces image of a sample by scanning it with a focused beam of electrons. The electrons interact with the atoms in the sample, producing various signals known as secondary electrons that contain information about the sample's surface topography and composition. SEM can achieve resolution better than 1 nanometer. Specimens can be observed in high vacuum, low vacuum, in wet conditions, and a wide range of cryogenic or elevated temperatures. Principle. The basic principle is that a beam of electrons is generated by a suitable source, typically a tungsten filament or a field emission gun. The electron microscope is accelerated through a high voltage, that is 20 kilovolt, and passed through a system of apertures and electromagnetic lenses to produce a thin beam of electrons. Then the beam scans the surface of specimen. Electrons are emitted from the specimen by the action of scanning beam and collected by a suitably positioned detector. Here's a diagram of scanning electron microscopy. Moving towards its construction, scanning electron microscope's basic components are electron gun, also called filament, condenser lenses, objective aperture, scan coils, chamber, detectors, computer hardware, and software. Electron guns. Electron guns are typically one of two types. First, thermionic guns, and second, field emission guns. First of all, thermionic guns. Thermionic guns are the most common type. Apply thermal energy to a filament to coax electrons away from the gun and toward the specimens under examination, usually made of tungsten, which has a high melting point. Next is the field emission guns that create a strong electrical field to pull electrons away from the atoms they are associated with. Electron guns are located either at the very top or at very bottom of a SEM and fire a beam of electrons at the object under examination. Condenser lenses. Just like optical microscopes, SEMs use condenser lenses to produce clear and detailed images. Instead, the condenser lenses are made of magnets capable of bending the path of electrons. By doing so, the condenser lenses focus and control the electron beam, ensuring that the electrons end up precisely where they need to go. Objective aperture. The objective aperture arm fits above the objective lenses in the SEM. It is a metal rod that holds a thin plate of metal containing 4 holes. Over this fits a much thinner rectangle of metal with holes of different sizes. By moving the arm in and out, different sized holes can be put into the beam path. An aperture holder. This arm holds a thin metal strip with different sized holes that line up with the larger holes. The metal strip is called an aperture strip. The aperture stops electrons that are off axis or off energy from progressing down the column. It can also narrow the beam below the aperture depending on the size of the hole selected. Here are the 3 diagrams shown. Firstly, the sample is at focus. First stage shows the sample at focus. Next one is over focus, and third one is for under focus sample. Scan coils. The scanning coils consist of two solenoids oriented in such a way as to create two magnetic fields perpendicular to each other. Varying the current in one solenoid causes the electrons to move left to right. Varying the current in other solenoid forces these electrons to move at right angles to this direction and downwards. Chamber. The sample chamber of SEM is where researchers place the specimen that they are examining. Because the specimen must be kept extremely still for the microscope to produce clear images, the sample chamber must be very sturdy and insulated from vibration. In fact, SEMs are so sensitive to vibrations that they are often installed on the ground floor of a building. They are also manipulated the specimen, placing it at different angles and moving it so that researchers don't have to constantly remount the object to take different images. Detectors. SEM's various types of detectors as the eyes of the microscope. The device detect the various ways that the electron beam interacts with the sample object. For instance, Everhard Thornley detectors register secondary electrons, which are electrons dislodged from the outer surface of a specimen. These detectors are capable of producing the most detailed images of an object's surface. Other detectors, such as backscattered electron detectors and X-ray detectors, can tell researchers about the composition of a substance. Vacuum chamber. SEMs require a vacuum to operate. Without a vacuum, the electron beam generated by electron gun would encounter constant interference from the air particles in the atmosphere. Not only would these particles block the path of the electron beam, they would also be knocked out of the air and onto the specimen, which would distort the surface of the specimen. Here is a diagram showing parts of a scanning electron microscope. First of all, electron gun releases the electrons, then they are accelerated up to dozens of kilowatt energy focused into the electromagnetic lenses injected to the surface of the specimen. Then a detector is installed which detects signals, and image is developed by synchronizing and the amplified current signal and the monitor. Applications. SEMs have a variety of applications in a number of scientific and industrially related fields, especially where characterizations of solid material is beneficial. In addition to topographical, morphological, and compositional information, a scanning electron microscope can detect and analyze surface fractures, provide information in microstructures, examine surface contaminations, reveal spatial variations in chemical compositions, provide qualitative chemical analysis, and identify crystalline structures. In addition, SEMs have a practical, industrial, and technological applications such as semiconductors inspection, production line of municipal products, and assembly of microchips for computers. SEMs can be as essential research tool in fields such as life science, biology, gemology, medical and forensic science, metallurgy. Disadvantages. SEMs are expensive, large, and must be housed in an area free of any possible electric, magnetic, or vibration interference. Maintenance involves keeping a steady voltage, currents to electromagnetic coils, and circulation of cool water. SEMs are limited to solid inorganic samples, small enough to fit inside the vacuum chamber that can handle moderate vacuum pressure. Here some images are shown from SEM, pollen grains of some plants, retina rods and cones, seeding coating, moth, and blood clot. And here's bacteriophage diagram. That's all related to scanning electron microscope. Thank you.